Professor Bruce Caswell from Engineering at Brown. We'll speak about dissipative particle dynamics, simulations of red blood cells, and their suspensions in health and disease.
Professor George Karniadakis, with whom I work, has been engaged in a project of simulating the circulation in the human body for perhaps 15 years or more. And that project, in fact, is part of a global project to construct what is called the virtual human, where every organ in the body is to be modeled, and then they put the whole thing together. Hopefully it will behave something like the real thing. But anyway, we're some ways off that. So what you see in this slide, I'm going to be talking about the dissipative particle dynamics method, which is appropriate to the simulation of cells, things at that level. And what this box shows is what dissipative particle dynamics is all about. We'll have, in this case, just a polymer chain represented by the red dots, which are just particles joined by springs, and the sort of pepper, the black particles represent the solvent. And if you know anything about fluids, you might wonder how so few particles could represent a liquid, but we'll see that they do. And I hope to convince you of that, at least as we go along. On the left, you have a picture of normal red cells, probably with some platelets, the yellow pieces showing also. On the right, you have a picture of malaria-infected cells. The one at the back is full of parasites, and they're still contained within it. Inside the red cell, the immune system doesn't know they're there, so they stay in. Eventually, they break out, as the second one shows, and they have to quickly find another red cell to burrow into. Otherwise, the immune system will take them out. Now, I should mention we have a number of students who have been working on this. Juan Lei and Shuijin Li, who are still at Brown. Dmitry Fedosov, who did a great deal of the work I'm talking about here, is now in Germany at Jülich. And Wen Xiaopan, who is up north in the Pacific National Nuclear Labs in the state of Washington. The experimental work that we are often trying to model is being done at MIT. The lab was originally set up by Subra Suresh, of course, who's now at the head of NSF, and the lab is now being supervised by Ming Dao.
Bruce, is that a particular infection, or is this a very general picture?
Of malaria?
Right.
Yes, that's one of the last stages of malaria.
Malaria.
Yes, malaria. Yeah, we'll see. But that's what the main infection I'm going to be talking about is malaria. Under the influence of the Brownian motion, that's an equilibrium simulation. It was done. There are about 4,000 particles in the box, and that calculation was done about 5 years ago on a workstation. So you can do a little bit of DPD with relatively modest resources. Now, not only the circulation, but there's this particular sample of arteries has a huge aneurysm, and we have also modeled the deposition of platelets. And I'll go on and talk about the main part of my talk. What is the motivation for looking at biomechanical blood components? Well, there are diseases like malaria where in the small vessels, because of changes in cell properties especially, you get occlusion, which is just a way of saying reduced flow, and actually jamming and blocking. So we try to model that. Examples are particularly malaria, and sickle cell, of course, is not an infectious disease, but it is a consequence of malaria. That is, it's an adaptation, a negative adaptation to malaria, and so is elliptocytosis, which I won't talk about at all. You're much worse off having sickle cell, I think, than malaria. We know from recent announcements that there's a hope that with malaria there will be a vaccine might be helpful. And so we will talk about dissipative particle dynamics for a little bit when we discuss malaria a little. Malaria cases occur annually worldwide at the rate of about 3.5 to 500 million. Of these, about 1 to 3 million result in death, the majority of being children. And this is considered a major hindrance in some countries to economic development. I think that's the only time I'll mention anything to do with the social sciences. You can see from the map here that the places that are particularly affected, Africa and South America, but also Asia. And in some of these African countries in West Africa, the incidence is about 1/4 of the population has been infected. So to understand malaria infection, you start off with a mosquito bite, which injects the parasite into the bloodstream, and it makes its way to the liver if it doesn't get done in by the immune system. There it develops into a different form, and I won't bother with the names, and leaves the liver and goes into the bloodstream and searches out red cells in which to hide from the immune system and to grow, multiply by an asexual method, because in fact these parasites are a male and female. In order to continue their life, the mosquito has to bite you again, and they pick up the malarial cells, and the reproduction, the sexual reproduction, actually occurs in the mosquito, which then re-injects them into a person. One of the features of malarial cells is, unlike ordinary red cells, normal ones, they adhere to the cell walls. We'll see how that helps the malaria cell survive the processes that go on inside the body.
Before you go back, one last one, where's Haiti?
Have you heard it?
Haiti?
No.
They have quite a bit of malaria right now.
Yes, I think it's probably too small to show. There's a small dot there.
Yeah.
That may be Haiti. Yeah.
Oh, yeah, I see it.
Yeah. This set of experiments in microchannels, being plaques that are very closely spaced, probably about 4 microns spacing between them, and you're looking at the plane of the microchannel where there is a constriction to the flow. In each case, the flow is from right to left, and the column here, this column describes the stages of malaria. So rings, in the ring stage, you have about 1 bug per cell, and then they grow in number until you get to the croissants, where you have a dozen to a dozen and a half inside. This red line separates out total blockage. In other words, all the squares below the red line, there is total blockage. So you can see in the ring stage, the cells can still get through the very narrow passages. You have to remember the 8 microns, the A column, the A at the top here, that channel corresponds to the size of a red cell. The diameter of a red cell is 8 microns. So all these are smaller, and the capillaries very typically are smaller than the red cell. A normal red cell has to fold up to traverse the capillary system. So that gives you an idea of what happens is you're going to get blockage. And when you have blockage, of course, there are very severe consequences to follow. Not only the obvious one, which is strokes, but in the other organs of the body, you can also get very similar things. Now, here's one of the problems we have with modeling. You have to decide where you are. If you're in the main arteries, like the aorta, you can get by with a one-dimensional model, which I would say is not so different from the hydraulics model, which engineers use to model pipes. You have to take into account the pulsatile nature of the flow and the flexibility of the channel. When you go into the smaller arteries, you can do that until you come to a junction. Of course, there are almost 60 major junctions in the arterial tree, so you have to then use the 3D continuum Navier-Stokes equations to do that, and you have to find ways of joining these solutions together, and all that has been done. So when you get down into the red blood cells and the capillaries and the venues, this is the domain of dissipative particle dynamics. It's a mesoscopic scale system. Molecular dynamics takes you down into the atomic level, and that is not what we need. Here's another thing which says more or less the same thing. Dissipative particle dynamics was started about 1992 by Hugenbrugge and Coleman, and its domain lies, as I said before, between the atomistic level and the continuum level. It can be considered to be momentum-conserving Brownian dynamics. If you think of Einstein invented what is now called Brownian dynamics. He considered a sphere, which was a pollen particle, in a fluctuating continuous fluid. In our case, the fluid is treated as a particulate system rather than a continuum. That's the main difference between those two type things. The triple-decker system, which you would have seen an example of in that movie, had it worked, is a way of splicing these levels together. In fact, here is a description of it. If you take a domain and you decide that you can use the Navier-Stokes equations in part of it, and DPD and MD and other parts of it, how do you put together those solutions so that they splice together or they match appropriately?
How do you?
Well, I don't think I'm going to go into all the details, but that is published. Here's an example. This is the classic problem in numerical testing of numerical schemes for the Navier-Stokes equations. You have this cavity, square cavity, and the top of it is sliding across at velocity v. Now the problem for the Navier-Stokes equations here is that at these points where the stationary walls meet the moving plate, there is a singularity in the solution which is not integrable, which means if you have a finite difference method and you keep refining your mesh, your answer, you're going to run out of digits in your computer because the numbers there are going to get so large that you won't be able to handle it. So one way out is to treat that to a molecular dynamics scheme and a DPD scheme somewhere in between, and then the Navier-Stokes. And here is a result. Of course, this is the velocity, and it's not, of course, near the top plate where the singularity is, but it compares the Navier-Stokes predictions to what they obtained by this overlapping of these three methods. There's a further example. So what we do in dissipative particle dynamics, it's really, in principle, very simple. Each particle satisfies Newton's laws, so we have to decide what the forces are. So the total force is typically a conservative force, a random force, and a dissipative force. The reason you need the dissipative force is when you put in the random force, and you can see that it's a stochastic term with this square root in here. The random force puts energy into the system, so the dissipative force takes it out. And surprisingly, the conservative force is a linear function of the distance from the center. So the particles are point centers of repulsion with a linear distribution of force down to some cutoff. The random force has a random variable, as you would expect, this c. The Rij is as a vector would be the vector connecting any two particles. So we're only considering binary interactions. So the random force has this function W sub r in it, where p is something you have to choose. It's usually between about 1/4 and 1. And the dissipative force involves the velocity differences. Vij is the difference velocity between any two particles. So the fluctuation dissipation theorem, which must be satisfied in order to bring temperature into the problem, forces WD and WR to be related in this simple fashion by the square, and the constants which characterize these forces, sigma and gamma, to be related in this fashion. So that brings in temperature, and that is your thermostat, which is a necessity to make the thing work. Now, how can we justify this DPD? It seems like an arbitrary procedure. Well, one of the ways is that we've done, and other people have done similar things. I think we've removed some of the restrictions on them, is that initially you take some simple atoms like Leonard-Jones particles. So you consider points and the interactions, they interact through Leonard-Jones potentials, and that's your MD system. And you group them into spheres, and the radius of gyration, which is the square root of the second moment of those particle positions, you take to be a constant. Now, these spheres can pass through each other. It's just that somehow or other these particles must stay within a certain position relative to each other by this constraint. As you're doing the simulation, you regard the center of mass of each of these spheres as one of these DPD particles, which we can look at here. And so you can extract certain information like the forces that are acting on the center of mass. And you can also compare the predictions for macroscopic properties like the viscosity and the coefficient of self-diffusion to find consistency. And what we find here is that when we look at the conservative, the main part of the force, the conservative part, which we're plotting here as a potential rather than as the force, beta here is 1 over kT. So we find that if we vary the temperature, the absolute temperature, by more than a factor of 2, as long as Rg is greater than 1, we can find where the units of Rg here are atomistic, that they would be at the Leonard-Jones level. Then these curves collapse and then we get an entirely repulsive potential, and it has the hallmarks of a mesoscopic quantity. The theoretical known theories of mesoscopic systems, like the Langevin formula for the force on a polymer, a tethered polymer, that is always proportional to kT and the number of particles. If your Rg is a bit too tight, then you still get some attraction in your potential, and you don't get the collapse into a common set of curves. We can also distinguish the dissipative force out of this and find the coefficients of the dissipative force. Now, in the original slide, you probably didn't notice, but the dissipative force was taken to be central, that is, lined up along the distance between the particles. What you find from this study is it's generally not true. It doesn't show too much here because of the initial scale here involves the startup of the system. The DPD system would be valid at longer time, since this time is on the atomic level. So if you were to plot this on a much smaller scale here, then you would see that there's a distinct difference between the parallel and the perpendicular force coefficients. So in some problems, once you get into that, you have to take into account angular momentum, and we have done that in some cases, but I won't dwell on that too much. Now, what about modeling red cells? Well, we've developed two models. One we call the multi-scale model, which I'll try and explain why we call it that. The other one's a low-dimensional one. The advantage of this one is it's not quite so accurate, but it costs about 50 times less to run. So we try to fit all the parameters with some experiments and predict the outcomes of other experiments. In other words, we try to have models which really have some predictive power. In the case of malaria, we have to add in wall adhesion forces. When we form suspensions, there are other forces between particles that we have to add, which are attractive forces to simulate the reversible aggregation and disaggregation of red cells into Rouleaux. Under static or near static conditions, blood cells tend to stack up like coins or poker chips. The French word for that is Rouleaux. So one of the things we'll do is predict the viscosity in Colloid flow and look at quasi flow, and we won't change the model at all. We'll keep the same parameters. So what is we have to model? Well, this is I guess a cartoon of a red cell. Here are the major dimensions. There are something like 27 to 40,000 junctions in this spectrum skeleton, which underlies a lipid bilayer. The lipid bilayer is essentially area incompressible. So that's something we have to take account of. And it's the spectrum, of course, which gives the cell its elasticity. And there are junctions between these two things. These are band 3 is a junction protein. However, we're not going to model that in anything like that detail. So let's look and see. Let's see if this works. Oops. I'm trying to get another movie going here. I don't seem to be having much success with movies. Oh, here it goes. This is our red cell model, and it's at equilibrium, and it's moving because of the Brownian motion. So in this model, every vertex is a DPD particle, and each edge is a viscoelastic spring, which consists of what's called the worm-like chain, which is, we've got KT in front of it, as I mentioned. It's a mesoscopic type of entity, but it's derivable from statistical mechanics. Now we've added a compressive term here because we find that that this model, the worm-like chain, like many others, when the force is 0, the extension is 0. So what happens then is these particles, when they're unloaded, will simply move together and you'll get kinks, artificial kinks. So you put a compressive force here so that at 0 extension, you have a finite length. So we also have to add in bending forces. It's a very common way of treating bending in particulate systems. So each of those triangular plaques has a bending constraint in it. The other thing we have to do is to keep the area of each of these triangles constant, because it's the lipid bilayer is essentially area incompressible. And of course the interior cytosol we treat as an incompressible fluid. Now, in order to describe our low-dimensional model, I'll take a brief detour into our treatment of colloidal particles. On the left here, you'll see the force, the interparticle force, represented by an exponential formula. And by varying the parameter b, we can either create a very hard core, which is the upper curve, a very soft potential, which is the lower one, or the linear one, which is the one we've already seen. So for the colloid-colloid interactions, we pick the upper one. Colloid solvent, we pick the soft one, and solvent-solvent, we pick the usual one. And you can see the radial distribution function that we get at various volume fractions of this colloid always give you this picture, which is very typical of hard spheres. And when we calculate the viscosity here, we get results, the red dots, which are very much in agreement with Bisserano's formula, which is considered to be the best representation of the data and the theories. Now you'll notice we also did the open stars, red stars, when it says standard DPD, we use central dissipative forces in that case, and it's not quite so good. The solid red dots we've taken into account the angular momentum, and therefore the dissipative forces are non-parallel. So because these are point particles, we can put them together, and that's how we constructed our low-dimensional model. So the centers are these dots, and they're connected again by worm-like chain springs, and there's also bending energy to make sure they work right. So this is just another picture of a single colloid particle. You can see the solvent interacting right near the boundary, and then the solvent concentration becomes constant. Now, how do we measure how do we characterize these models? I mean, they've got parameters in them, so how do we measure things? Well, I guess we can't. Can this thing be pulled down at all?
I think so. No, I think it's the screen.
Anyway, never mind. These data come from the MIT people. And basically what is done is that a red cell in the middle is taken and two microspheres of polystyrene are stuck hopefully along a diameter. It looks like they didn't quite make it on that one. And then you apply laser light pressure on the spheres and that pulls out the cell. And from that you can construct a diagram of force versus of diameters versus force. So you have the major axis and the transverse axis. And the transverse case doesn't fit the data quite so well. And we think that's because of the difficulty of putting these spheres exactly along a diameter. Anyway, this is where the name multiscale comes from, because whether we have 100 particles to describe the cell, as in this picture here, or we have 27,000, which is the spectrum level, we get just about the same fit. So normally when we're doing our calculations, we pick 500. If we're going to distort the cells excessively, then we pick more. And these are typical numbers for the shear modulus, Young's modulus, and the bending modulus that is used. This test is not very sensitive to the bending modulus, so we have to look at other things. So in malaria, here are the stages of malaria. This is normal, this is healthy, and two malarial cases. And what happens, you can see, is that the extension is much smaller for the same force because the cells are getting stiffer. And you can see that in the diagrams here.
So just to clarify, you're actually experimenting there with malaria cells?
Yes, those are malarial.
And you got those from a donor or whatever?
Yes, this is published with the Suresh and Company.
And just for asking a question, earlier where you had your multi-scale, you had a certain scale of variance actually, with your curve. What causes that scale of variance? Do you know? Right?
Yeah, we don't know. That's why we call it.
Oh, okay. Okay.
That's why we call it what we say it's multi-scale, because it's insensitive. It wasn't always that way. Before, I mentioned that we have a compressive term in the spring to give us a finite size when there's no force. Before we did that, we had a lot of kinks. These curves were very sensitive to the numbers.
So therefore,
Yeah. Yeah, that took a little working out. Okay, so what you can see in the malarial case is that there's definitely a big increase in the stiffness of the cells. And you can see from the modulus, which is shown here, it grows by almost a factor of 10 before you're dead, so to speak. See if I can get this to work. Yes. What happens if you put a single cell into a shear flow? So you have a gap, and you're shearing it, and you put the cell more or less in the center of it. At lower shear rates, the cell will tumble and not distort very much. Oops. At the higher rates, it will tank tread. So the cell isn't really rotating very much, but the outer skin is going around and around like the treads of a tank. Now what these curves show us is the following. If we reduce the viscosity of both the suspending fluid and the inner fluid to practically zero, which we can do with our DPD system, then we get the transition from tumbling to tank treading is this line, and we get this line, we get the black line. If we add in the viscosities of the two fluids, we get the green line. But we also have to add in the viscosity of the membrane. The membrane has is a viscoelastic thing. So when we add that in, we get pretty good agreement with the experiment. One of the things that the tumbling case also helped us with is to get the bending modulus fairly correctly. So if I can never see, oh here it is, I should have my glasses on, I guess. This is an experiment which is called magnetic twisting cytometry. You adhere a cell to a substrate, and then you stick a magnetic sphere on the surface, and then you drive it magnetically. So you get, what you're doing is getting the frequency response, which is what these wiggles tell you. And from that, you can deduce the viscoelastic modulus to within a constant, because the geometry is so complicated, you don't know that constant. If you had a plane half body, there are those, engineers do those indentation problems where they press a sphere into a half body, and there's a lot of mathematics, and they get some answers out of it. Well, this geometry is much too complicated to do that with, but when we look at the data, if we slide, because we know G and G primed independently, if we make one of them agree, then the second one agrees, provided we have the bending modulus reasonably well established. Of course, this is for healthy cells. We don't do so well when we look at the case of the malaria infected ones. In the first place, we only have one frequency in the experiment, so we don't know too well. But they're not, G primed are the horizontal lines, and they don't agree very well. So one of the things we don't know here is, of course, what's going on. All we know is we're characterizing the malaria cells with their stretching properties, and we don't know what the role of the parasites are. So that's really probably, and we don't know how the membrane viscosity changes because we don't have enough data. Now, if we want to do suspensions, we have to put some attractive forces, which we do in the form of a Morse potential. Morse potential looks like the Leonard-Jones. It has a minimum net, and you therefore get an attraction, which we have to tune. We try to tune the forces so they are much smaller than the forces required to distort the cells, because when you look at pictures of Rouleaux, the cells don't look distorted very much. So that's how we got them, and in this case you see the low shear on the left. You can see there are some structures forming in there, and they're breaking up at high shear rates. So what do we do with that? We measure the viscosity in a uniform shear flow, Couette flow. That's the only way you can think about viscosity of particulates like blood, because the concentration remains uniform. So what do we have here? We have two curves. One of them is whole blood, and it rises as the shear rate, which is what's on the horizontal axis here. In reciprocal seconds becomes small. The relative viscosity is the viscosity divided by the viscosity of the plasma. Plasma viscosity varies by up to 15%, so that tends to normalize things. The dots are measurements from at least 3 different laboratories over a period of about 20 years, starting in the 1960s. The second one, that's whole blood. The only thing that's been done to it is to add an anticoagulant, since you can't work with blood unless you do that. The lower curve is the curve you get for washed erythrocytes, red blood cells. You take a blood sample, you spin it down, separate out the plasma, and you resuspend those cells in some varying kinds of agents which strip off all the sticky stuff. So when you do that and resuspend it, then you get the lower curve, and the blue lines are our low-dimensional model, and the black lines are the multi-scale model. This is for male blood. Female blood is more like 40%, so you would get different curves because, of course, the viscosity is very sensitive to the concentration. Here's a picture of some actual Rouleaux. Here's a picture of what the red cells look like in real case when you're at low shear rates, and at high shear rates you can see their elongation, which is very much like the model gives you. Up in this little graph on the right here, we have an example of malaria infected blood. It's not tube flow, even though the picture looks like it is. The blue cells are the malarial cells, and the viscosity is measured at a given shear rate, and the percentage of malarial cells is varied. So from our model parameters, we predict this red line without any further adjustment of parameters.
So, sorry, do you see Rouleaux in your simulations?
Beg pardon?
Do you see Rouleaux in your simulations?
Well, it's hard to look at. Supposedly you can see them here, but you know, I agree that that doesn't look very convincing. Yes, we do. I mean, if we go back to
But
the,
not, so,
if we go back to this one, where are we? Here we are. There are some Rouleaux here. There's a beginning of a Rouleaux. If we lower the shear rate sufficiently, they will, in these tests, they will form up into stacks of about 20. They actually will form up.
if we go to the SEM picture. Now, so figure A there, do you ever see anything like that, or that's kind of artificial?
No, this is the real case.
Yeah, but it's fixed or something, probably.
Hmm?
It's probably...
Well, it's probably done under it's done under a microscope, so
That's kind of why I asked. None of yours look like that.
not quite like that, no.
Not at all like that, because that's like stacks of coins in
Yeah.
vacuum.
Nonetheless, we account for this rise in viscosity.
That's where I was going. So is that picture A that they show misleading? It's really much more
No,
distorted the way
these photographs are taken from a book. And they're just typical cases. I don't know. I couldn't even find out what shear rate
I guess,
these corresponded.
but in vivo, would you see Rouleaux to like the micrograph labeled A, or?
I think so, but it's quite difficult to look at them. That's the problem with, whenever you try to do anything in vivo, it's quite difficult.
Right.
There's a doctor in Montreal, I'm trying to think of his name, who is trying to spot Rouleaux in vivo by using ultrasound. And we're thinking of trying to simulate that. But anyway, that's the direction it seems to go, because there are a lot of people are not quite sure in disease exactly what happens to these Rouleaux. As soon as you start moving, if you're sitting as you are, you probably have Rouleaux somewhere inside your small vessels. If I fire off a revolver, you'll jump into action and you'll stop. They will be destroyed immediately. All the animals that have been tested, all those which have a fast response, like antelopes, and also the things that prey on antelopes, like the big cats, the rise in their viscosity and low shear rate is much, much bigger than this. Cows, they're much more like this. Either that's been bred out of them through domestication or never existed. I don't know which.
And on the inset, on the upper right,
Yeah.
I didn't understand the viscosities. Those are in the 2 range, but viscosity of blood is 3 or higher.
Beg your pardon? The dots are the experiments. The line is the numerical simulation.
Right, and the values in centipoise are like 2.
Yeah,
But blood has a viscosity 3 or higher, right?
that's probably erroneous, that chart. Yes, I think you're probably right. I think this is meant to be the relative viscosity. That's what it is.
Yeah, but even then, the lowest relative viscosity is around 3 relative to plasma. Plasma is 1.2
Yeah.
plus.
No, you're right. You're right. I hadn't noticed that before. I know who made that graph, and I'll get after him when I get back. When we measure the shear stress from which we derive the viscosity, we can also measure the normal stresses. The only interesting thing about the normal stresses, I think, is that they've never been measured because it's too difficult. Blood has too low a viscosity, so when you put it into one of those rotational instruments that are used to measure normal stresses, you simply measure inertia effects. We've also calculated the effective concentration. Shin measured the concentration effect and the relative viscosity at various constant shear rates, and he made a nice correlation of it. So you can see that we predict that quite well with our low-dimensional model. Now, what happens in quasi-flow? And things are quite different. The hematocrit, which in shear flow is uniform in space, becomes highly non-uniform, and these lines are sort of ragged because we don't have that many cells, so you have statistical problems in getting a nice distribution. The velocity distributions are smooth because it's the velocity of all the particles. It's the mean velocity. And unfortunately, we can't see the bottoms of these curves. For the larger tubes of this series, we went from about 10 microns to 150. And you do get a flat portion, but you still get a peak in the middle. And of course, you have a layer near the wall called the cell-free layer where there's nothing in both cases. And that cell-free layer seems to grow as you get to smaller tubes. Although ultimately, when you get down to capillary size, the cells rub up against it, so this idea loses force. What's worth noting here is that the dashed curve is a parabola which has been fitted, and it just about fits everything exactly except at the wall. The straight line is what you predict if you use the wall, the solvent viscosity in that region. Now, one of the things we can do is take a slice through the tube and look at the cells and their distribution visually. We can also measure the asphericity. The asphericity is, not surprisingly, is defined to be 0 for spheres. It's also derived from the second moment of area. And for cells at equilibrium, their distribution of asphericity is this vertical line. It's about 0.16 or something like that. And the cells near the center line pretty well fall on that distribution. If you move outwards from the center line, then the asphericity gets larger, as you can see in both cases. Now, one of the interesting things that happens that we found out was that because in a quasi flow, you know the shear stress at every position R. The wall stress is the radius, the density, and the driving force, which continuing to be the pressure gradient. So once you know, once you have that, you know the stress everywhere. So if you differentiate the velocity curves, you get a shear rate, and that's this plot here. And therefore, if you know the stress and the shear rate, you can get the viscosity, which is plotted here. And you can see that our calculated viscosities, local viscosities, agree with measurements at those particular shear rates. Those measurements being in uniform shear flow. All this plot shows you is what happens if you put aggregation into the cells. The previous cases, we left out the aggregation. So what happens is that you get, as you might expect, more cells near the center line. In fact, you get an off-center peak. Off-center peaks have been measured. The measurements are very sort of imprecise, but you can see that effect. And the velocity profiles are not very insensitive to that. There, in tube flow, you get involved with two kinds of measurements of the hematocrit. There's the tube hematocrit, which is just the average number of cells in a given volume of the tube at any that you put in to start with. The discharge hematocrit comes from integrating the concentration times the velocity. It's also the feed hematocrit. The reason, of course, that you have different ones is that there's this cell-free layer. We find its distribution. We find the distribution of the edges of the cells, and we plot them into distribution, and the peak of the distribution we take to be delta. So we have a few measurements of the three curves. I won't explain the upper and the lower curve, but the main curve that we calculated is, as in the previous slide, is the middle one, and we have a few measurements, one of them at a different hematocrit, but they're still not the range that we expect. And if we do that for malaria, where we don't have any measurements, we find that we have decreased the cell-free layer, but we're not including perhaps some effects that are important in malaria. We can calculate the flow resistance, which people who measure blood flow in tubes like to report as an apparent viscosity. The viscosity you would get from using Poisson's law, in this case divided by the solvent viscosity. And they plot that versus diameter, so that there's a correlation of Pries, and that's this curve. and the dots here are numerical.
Do you think you'll ever see it going back up there at the very small diameter?
Well, if we have gone to about, we have gone a little lower than that. They're not on this plot. I disregard this other plot here. It's a different set of variables, but the apparent viscosity versus diameter. Experimentally, people have done that. Yeah.
And that's just having trouble squeezing through the channel?
Right, right. Once you have to squeeze. Oh, here is. Let's see if the movie works. Yes, it's going to work. That's what happens to a blood cell when it gets into a small tube. It folds up into this mushroom cap shape. So there's a lot of resistance there. So that brings us to another feature of flowing tubes and small tubes. On the epithelium, which is the layer cells which line the blood vessels, is something called the Glycocalyx. And if you don't want to remember that name, it's easier to remember it as slime. When you catch a fish, you haul it in on your rod and you take hold of it, and you know that if you're not careful while you're pulling out the hook, the fish will slip away. What's on the surface of the fish is a glycocalyx layer, perhaps not the same as the one that's in the blood vessels, but nonetheless that's what it is. The yellow dots here are the solvent particles, and the glycocalyx here is represented by very flexible beams. The real one is like a tree trunk with branches. But this is the way we've modeled it so far. I don't have a movie of this, but you can see the red cells are rubbing up against the glycocalyx rather than the wall. The glycocalyx is important this way. Damage to the glycocalyx is always associated with inflammation. So that's something that's of interest. And if we want to get to blood flow in vivo, this is something we have to do. Notice here there are some cells sticking to the walls. This channel, the walls have been coated with this stuff to mimic the epithelium. And these cells that are white are the malarial cells. The other ones are normal ones. And see if we can get the movie to work. That's the trick. And you'll see this, the red cell, the malaria cells are flipping along the surface. So
That's a simulated vessel?
no, this is an actual experiment. This is a simulation. Here we're not showing the red cells, just the diseased cells. You can see they flip along quite nicely.
Can I ask a question about that?
Yeah.
Forgive my vulgar functionalism, but is there a benefit to the parasite side for adhering to the...
Yes, because if I might run out of time, because otherwise it will be trapped by being dragged into the spleen. The spleen has very narrow passages, and any stiff cells get destroyed. Your red cells normally last 120 days. As they age, they get stiffer, and eventually that's one of the places where they get removed. So any parasitized cells that get into the spleen won't get out. So if they could, then that would be the end of it. You wouldn't. In fact, here's our adhesion model. These black lines, ligands, you're joining the cell to the surface, it's a stochastic model. They have a formation rate which depends on the elastic energy and a dissociation rate from which we can construct a probability. And that's how we get this thing. I'm going to skip the next two because of time. Let's see if we can get this movie to run. Probably can't. Nope. It's one of those that won't. We actually measured the rate at which these things move along the wall, and our model does it. It's the black line here. Does a fairly good job of it. And the mean value is this blue line, experimental curve. At very large stresses, it doesn't work so well. Now here is in fact this experimental spleen. So we have a channel, 4 microns gap. The posts, these little objects, which are shaped like triangles, in this case like lozenges, are about 4 microns high, and the spacing between them in the horizontal plane is typically 3 microns, and those are about the spacings of the spleen. So I don't have the movie here, but since we're running out of time, it's probably just as well. Let's see if this movie will work. Yes, here, this is our simulation, and you can see how distorted the cell is in each of these cases. It's very highly distorted, so if the cell is at all stiff, it just won't make it through those little spaces. In fact, when we look at measurements in that device, then we find that the ring, the first stage of malaria, they can get through these obstacles, but at about half the rate of normal cells. Not surprisingly, it's easier to get through converging than diverging geometries. What we're working on now is sickle cell. We've learned how to create these oddball shapes which sickle cells get into, and we have viscosity measurements, and they're not too bad considering the simplicity of what we haven't put anything inside the sickle cell, except that this is the beginnings of it. Sickle cell, the hemoglobin in sickle cell anemia is not normal. In the state of deoxygenated state, it polymerizes or gels, depending on how you want to describe it, and into rods and all kinds of odd-shaped things. So we've learned how to make these particles self-assemble in a box first and then inside a cell. So we go from the self-assembly process starts off this way, and by the time it's over, we've got something which looks like a sickle cell. There's a motivation for doing that, which I could describe if someone else asked me. So what can I say? We predict a certain number of things with our models. They're not perfect. For instance, we can't capture febrile temperature effects. We can do that for polymers, but in polymers you're changing the temperature by tens of degrees, which you can't obviously do. A 4 degree centigrade rise in temperature practically kills you. But we have looked at, the people at MIT have looked at how the properties of red cells change as the temperature increases, and we can't capture that. Presumably there are chemical changes going on which are not in the model. So we do a pretty good job on viscosity, and we find out some of the things that are going on in tube flow. So at the moment we're putting in the Glycocalyx, we're changing the tube geometry to include junctions, which are very important in the capillary system. And of course we always need more experimental data to check our models. So if anyone has any questions, I'd be glad to try to answer.
A lot of the problems you talk about, you're talking about the multi-scale and the numerical computations, etc. You know, there's problems on the boundaries as they used to be had. Now they're probably too technical, but give me at least a clue of how you have to see these things.
Yes. One of the things that happens with particle models of all kinds is that when you get near if you define a boundary to be a set of fixed particles, you line them up here, you say this is a boundary, and now you have your fluid particles rattling around in here. What happens is that you typically will find that you'll get density fluctuations which are not realistic. There's way more than you should have. So what we have to do in order to understand this is we put fake particles, one way is to put fake particles on the other side of the boundary and let them interact with the ones inside, and then try to adjust the stresses. We look at the pressure and the stresses at the same time to bring the two together. Some of that is published. I think I can spot if I look through my...
So in other words, what happens is that modeling, you're looking at precisely, in fact, a singularity type thing. So you're essentially extending the system by throwing in the antimatter.
That's one way of doing it, yeah. What we would do is we'd have a box here which contained, we'd let these things come to equilibrium and then we freeze them. But their forces of interaction can still...
But you got the repulsive and attractive force combination.
Yeah.
A 6-cloud type.
Yeah, I mean, it is somewhat empirical still. I don't think there's a clear theory of it. If you're interested in tough theoretical problems, that's one.
Those are difficult to go across almost all disciplines.
Yeah. Yes?
I learned a lot. Let me clarify a couple, just about how red blood cells work. I really enjoyed this. So how long did you say is the life of a red blood cell?
120 days is the typical number. The bone marrow is constantly generating.
And how often do they circulate through the body? Do most of them circulate more or less completely?
I'm trying to remember.
Stuck in organs sometimes.
Yeah, they go all the way around. One of the reasons they deteriorate, obviously, is they're forced into the capillaries. So I don't want to say it's like bending a piece of wire, which eventually breaks if you bend it enough times. But they do take a beating. And if they go through the spleen, it takes another beating.
So, much of the, in looking at malaria and sickle cell anemia, are there organs, however, where they do their damage and yet they circulate through the body? Is that?
Well, they get through some places and not others. I mean, in the case of sickle cell, the people suffering from it suffer terrible pain because it tends to affect all their organs. If it goes to your brain, then it probably, it might be the end. The children who die of malaria generally die because they suffer some kind of stroke. But the other organs are also, the blood circulation is reduced. It's just harder to get everything through if it does. And whole areas are depleted entirely of the circulation. So it's bad news.
So I'm trying to put the global picture.
I think if someone could find a way, for instance, of changing the adhesiveness of malaria, then that would be...
Seems like a nice, clear target.
Yes. But in the case of sickle, of course, it's worse because it's a hereditary disease. And there's no way you can change that as far as I know. There's only one treatment that is somewhat viable, but it sounds terrible to me. There's a chemotherapy agent called hydroxyurea, which is used to treat leukemia. Now, in leukemia, you give these agents to the patient hoping to induce a remission. And if you succeed, then you stop with that awful stuff. Well, one of the things that this hydroxyurea does is it induces the production of fetal hemoglobin. When you're a fetus, you have a different hemoglobin from when you're an adult. By the time you're 6 months old, the fetal hemoglobin has disappeared mostly, and you have adult hemoglobin. In fact, people with the sickle trait don't suffer until after that age, typically. And what fetal hemoglobin seems to do is it interferes with the gelation and setting up of these structures inside the cell. So the cell would stay softer and therefore would circulate more easily. The trouble with that is, of course, you've got to keep administering this dreadful stuff to these people. So
drinking lots of olive oil wouldn't help.
Yes, that's the way I would have thought about it. Apparently not.
There's a question over here.
So given what I said, I'm sort of thinking of possible applications for something like this. And there's one group in Seattle that's working on, so the big challenge is trying to figure out rapid diagnostic tests for malaria, particularly for one promising technology is to use lasers to look for hemoglobin, which is a crystal that's essentially generated by malaria parasites when sort of converting the heme, a byproduct into it into this crystalline form. Because otherwise it's harmful to them. Now this sort of thing seems, I mean, are these variations, so there are, for instance, I mean, I have an iPhone app which can check my pulse rate fairly accurately by essentially looking at just using the light of the camera to look at movements in the blood. So just off the finger, I can show it to you during the break. Are there sorts of things that might be able to measure viscosity and be able
in vivo, you mean?
Well, actually in a non-intrusive way, because right now
it's quite difficult. The problem in vivo is that you can get an estimate of the velocity, of the flow rate, but it's very difficult to measure pressure differences. You need both to measure the viscosity in a tubular flow. The other thing is the geometries. Generally, I mean, there aren't very many straight tubes in the circulatory system. You run into junctions and so on. So it's quite difficult to talk about viscosity in vivo. That is, and find something that's measurable.
Okay, but is there any manifestation of this that you think can be? I mean, the problem with the rapid diagnostic tests is that they're all invasive. They require, pricking the finger, which places with HIV and bloodborne pathogens. That's the problem. So I'm just trying to see if there's.
No, I can't think of anything. The Suresh had insisted that we together get a patent on the testing of these single cells combined with the simulations, because he thinks they may have a future as a way of certainly in research, a standardized way to characterize cells. I think most biomedicine is oriented towards biochemistry. People think in terms of a chemical agent, apart from things like bone fractures, obvious mechanical things. So if you have a disease, people think of curing it or treating it with chemical, biochemical. Whereas, and they measure. You have your blood tested, it's a chemical measurement. So much of this, so much of that. What he's arguing is that his mechanical tests actually tell you something about the condition of the blood.
Well, it certainly sounds like a promising avenue to look at. There might be things that are mechanical that can be done without use.
Quite possible. Well, ultrasound is one way to go.
Thank all the speakers.